morning everyone. Today I would like to present about the detection and enumeration of Listeria monocytogenes in food samples using fresh raw homogenized chicken. Firstly, Listeria is a genus of gram-positive, rod-shaped bacteria that are widely distributed in the environment, including soil, water, and decaying plant materials. It can also be found in various food products such as raw meat, processed meats, and ready-to-eat foods, as well as in the feces of humans and animals. In terms of morphology, Listeria monocytogenes is a gram-positive, rod-shaped, non-spore-forming, and facultatively anaerobic. It is also motile at room temperature due to the presence of flagella. These microorganisms can survive under harsh environmental conditions such as low temperatures and high salt concentrations. Because of this ability, Listeria monocytogenes can still grow in refrigerated foods, making it difficult to control in food industries. Among the species of Listeria, Listeria monocytogenes is the main pathogenic species that can cause foodborne illness known as listeriosis. Next, Listeria monocytogenes is commonly associated with ready-to-eat foods such as deli meats, smoked seafood, salads, and softies. These foods are considered high risk because they are usually consumed without further cooking. Cross-contamination can occur during food processing and handling through food contact surface, machinery, and workers. Post-processing recontamination is a major challenge. Environmental resilience. L-monocytogenes can grow at refrigeration temperature and survive under stress conditions like high salt concentration, making it difficult to control in cold stored foods. The presence of Listeria monocytogenes in food can cause listeriosis, which is a serious foodborne disease. Common symptoms include fever, nausea, diarrhea, and muscle pain. In severe case, it may lead to meningitis, septicemia, miscarriage in pregnant women, and even death in high-risk individuals. For this experiment, fresh raw chicken was used as the food sample. The sample was homogenized before analysis to ensure that the bacteria were evenly distributed throughout the sample. This is important to obtain accurate detection and enumeration results. Lastly, detection was carried out to confirm the presence of Listeria monocytogenes in food sample, while enumeration was performed to determine the number of bacterial colonies present.
On the first day, the homogeneous sample was added into UVM enrichment broth and was incubated for 24 hours. On the second day, after incubation, 1 ml of enriched culture was transferred into freezer broth. The freezer broth were now incubated again at 37 degrees Celsius for another 24 hours. Day 3, after incubation in freezer broth, the culture was treated onto Vulcan agar using the strip plate technique. Vulcan agar is a selective and differential medium commonly used for isolating Listeria species from food samples. The inoculated Peckham agar plate was then incubated at 37 degrees Celsius for 24 hours. On day 4, colonies suspected to be Listeria was selected from the Peckham agar plate and straight onto Tesaya agar to obtain pure culture. The Tesaya plate was then incubated again at 37 degrees Celsius. On the last day, biochemical tests were carried out to perform the presence of Listeria. The first test was the grain stain test to determine the grain reaction and cell morphology of the bacteria. First, a small amount of bacterial culture is smeared into a clean glass slide and allowed to air dry. After drying, the smear is heat fixed by gently passing a slide through a flame. Next slide is flooded with crystal violet stain for about 1 minute. After that, the slide is rinsed gently with water to remove excess stain. Then gram iodine is added for 1 minute. The slide is then washed again with water. The next step is decolorization process using alcohol. After decolorization, the slide is rinsed immediately with water to stop the reaction. Finally, the slide is counterstained with safranin for about 1 minute. The slide is then washed, dry, and observed under a microscope using immersion oil.
Firstly, for the Peckham agar result, we observe strong back colony growth in the positive control. This indicates the presence of Listeria species because Peckham agar is a selective medium where Listeria produce black or dark colonies. Next, on Tisei agar, there was strong white colony growth, showing that positive result. Tisei agar is used to support the growth of Listeria, and the appearance of smooth white colonies confirmed successful bacterial growth. For the gram staining result, observations were made under different magnification, which were 4, 10, 40, and 100 magnification. Under higher magnification, the cell appeared as purple box-shaped bacteria, which is characteristic of gram-positive Listeria species.
Based on our result, the positive control shows successful growth of Listeria on both Palkam agar and Tisai agar. For Palkam agar, strong black colony growth was observed, which indicates the presence of Listeria species because this medium is selective for Listeria. Law et al. State that all Listeria are capable of esculin hydrolysis, and this process will result in the formation of an intense black color in the media. On Tisai agar, strong white colony growth was also observed. This shows that the bacteria were able to grow well on the enrichment medium and confirms the viability of the culture. For gram staining, the bacteria appear as gram-positive cells under different magnifications. According to Rogala and Bomar, Listeria monocytogenes is a gram-positive that commonly found in soil, water, and vegetation. Overall, the results confirm that the methods used in this experiment were effective for detecting Listeria from raw chicken samples.
Angels up in the clouds, our jealous know